So our next speaker is Darach Miller from David Gresham's lab at NYU, and he's going to talk about genetic factors controlling accelerated mRNA degradation during a nitrogen upshift.
Okay. My name is Darach Miller, coming from the Gresham lab at NYU. That over there. I just want to thank the organizers, and especially Audrey Gash, for giving me a chance to get up here and tell you all about this work today. So I think Francois Jacob put it best, that the dream of every cell is to become two cells. I think that pretty much wraps it all up right there. But there's a catch, and that is, well, that yeast has to do so in conditions of feast and famine. And so it's evolved these conserved signaling pathways like TOR complex 1 or pKa that are able to sense limitations of nutrients and then regulate a coordinated program of growth that's appropriate for the particular nutrient limitation conditions. And so we study how nitrogen limitation affects growth and how nitrogen limitation thus is regulating this coordinated program of different growth rates. And so it's well known that one way in which yeast regulates its growth is through transcriptional regulation. So it's been demonstrated many times before, and thanks to the organizers for the excellent placing of the talks, is that this has been demonstrated many times before, is that about a quarter of the transcriptome is differently expressed, and that's just explained by changes in growth rate alone at steady state. I'm not talking about shifts in conditions right now, just steady state. And so it's not surprising then that in recent work, we've limited cells in chemostats for a variety of nitrogen sources, and we've been able to limit their growth on a variety of different nitrogen sources, different qualities of sources, at a variety of different rates. And what we found by ANCOVA analysis in collaboration with Ido Roldi is, again, that about a quarter of the transcriptome was differentially expressed, and that's well explained by just changes in growth rate. So it's not surprising then if we just take those samples of microarrays of chemostats of yeast cells that are just growing at different rates, and we plot them on the principal components of this data set, what we can see is that they're well separated just by changes in growth rate here. And so that's not surprising. But what I'm interested in doing here is just kind of motivating this is using these microarrays as kind of a snapshot of the regulatory state of the cell, because it's a very high dimensional and somewhat unbiased marker of where the cell is at in terms of its regulatory state. And what I want to do is start to look at how this changes as we vary growth rate. And the way we're going to do that is with a nitrogen upshift. And so here's an example of that in batch. So these are cells that are limited for growth. They only have a non-preferred source available, proline, and so they're doubling every 4.5 hours. We're using FI4, so it's growing really slowly. If I add a pulse of glutamine, they're eventually going to be reaching a new, more rapid growth rate after a period of physiologically remodeling, and they get fatter. But what I'm really interested in looking at is what happens in the transcriptome very early on. How does this transcriptome, in the broad sense, kind of reprogram for rapid growth? How does it shift from a slow growth state to a rapid growth state? Is this kind of a coordinated process? Stop using the, sorry, excuse me. And so what happens is very quickly, in 5, 10, and 30 minutes after adding glutamine to this cultures, you can see that the cells, at least in principal component state in this very broad sense, reach a kind of a state that's transient and very unlike either the beginning or the end of this upshift, before returning and then eventually looking far more like a more faster growing cell. And so this is also, you see a similar pattern with different nitrogen quality sources that we add. And so what we're interested in doing is studying this process and thinking about this as not just a transition between steady states, but thinking about the transient state that it enters into in order to reprogram for rapid growth. And so here's the general strategy. I already told you about the first one, which is that we're measuring transcriptomics during the upshift and using this kind of high dimensional marker as a snapshot of the regulatory state of the cell. Then we want to figure out what's contributing to that. And so what we're going to do is we're going to dial down to particular markers, transcripts that are changing that we think are indicative of the broad patterns of regulation. And then we've developed new tools to do genetic screens on the factors that are regulating particular transcript dynamics. And I'm going to tell you about that today. And then step 4 would be to go back to the top and see what those regulators do in the broad sense, but we haven't got there yet. So here are two main aspects that we think are really driving the broad sense reprogramming. One is the massive upregulation of the RP and RIBB transcripts, the ribosome protein ribosome biogenesis regulon. Here's just the average abundance of those regulons after time in the glutamine upshift. And so why that's interesting, how quick that is, that's in our paper that we just published, so I'm going to not talk about that, and I want to talk about the repression of the NCR. So this is a nitrogen catabolite repression regulon. These are transcripts that are important for the transport and metabolism of non-preferred or scarce nitrogen sources. And there's this really, really quick and transient repression of this regulon. And it's really quick, so we wanted to figure out how quick. Of course, you can change the abundance of mRNA by altering the synthesis rate or the degradation rate. And so a lot about the synthesis, right, the cradle of these NCR transcripts is, pretty well studied, so we want to know about the grave, about the degradation rate, and to see if that's a mechanism by which these are being repressed. So what we did is we just compared two numbers. We modeled the transcript abundance with respect to time to get a number for how fast they're going up or down, and then we compare that to a specific degradation rate from steady state, and this is using 4th uracil labeling to get it's a metabolic labeling approach, and we're using a protocol developed by my good friend and benchmate Benji Nemoitin as RateSeq in 2014. And so we just plot the two against each other. On the x-axis is, again, this rate of degradation at steady state, and on the y is the rate of decrease of the transcript. All the points that fall along the line or below are not terribly interesting. We can't say that much about them. But the ones that are falling above and largely above are ones that are decreasing faster than their steady state degradation rate, thus the degradation rate must be increased. And so what's curious is that in this experiment and also in batch upshift, going from proline to glutamine, this is in a chemostat, going from limitation to addition of glutamine, the 5 fastest decreasing transcripts are all these NCR transporters. And so we wanted to see if this is, is this real? So we turned to apply the labeling methodology from the X to the upshift experiment on the Y. And so what I mean by that is that we added 4th uracil, so we label the whole transcriptome. Then we add an excess of uracil to chase out that label, and then we interrupt that. And so we're just going to use qPCR to track GAP1 and DIP5. These guys right here, GAP1 and DIP5. And if we do that and we just add water at the dashed line here, it doesn't greatly affect it, and it takes about 45 minutes, 50 minutes for them to reach a new lower equilibrium. But if we add glutamine, we hasten the chase. And so what appears to be happening is that glutamine addition is destabilizing these transcripts. And so we thought this was kind of curious and really interesting. So from here on out, I'm just going to tell you about our work on GAP1 here and trying to figure out what's contributing to that degradation. And so this is really curious because we already know a lot about how GAP1 is subject to extensive translational synthesis control. So we also know that it's post-translationally controlled at the gene product. So what's going on here? Why another layer of control? And so there's been some ideas about this that have been refreshed with new insights from excellent studies at paired transcriptomics and proteomics, and such as also was just presented, looking at how what's going on with this. And so the thought is that, just to reiterate, is that accelerated mRNA degradation will allow you to focus a limited translational capacity on newly necessary transcripts. So these would be things like the RP-ribiregulon in this case. And so what we wanted to do is we did a genetic screen to find factors contributing to the wild-type mRNA dynamics because we wanted to find out of GAP1, because we wanted to find out what that is, and if we can break it, maybe we can see why this regulation. That was kind of the broad motivation. So, and it's in particular a pooled screen. So let me just walk through the general strategy here. So courtesy of collaboration with Charlie Boone, we got a deletion collection, non-essential, protrophic, haploid, and first thing we do is scrape it off the plate and we put it in a chemostat. We starve it for nitrogen, so the wild type will be expressing NCR and GAP1. We'll take a sample and then we'll add glutamine and then take another sample. And now we got to somehow sort these mutants out based on their GAP1 mRNA abundance. And we're going to do that using FACS. And so we first looked at what markers can we use of mRNA abundance in the cells to do this screen. And so we first looked at a protein reporter that's worked really well, like having a promoter driving GFP. And other people have had a lot of success with that. But we're looking at something where the transcript is gone within 10 minutes. And so the protein are operating at a completely different time scale. So that didn't work. But then we looked to an aptamer, and that wasn't quite bright enough. So then we looked to mRNA fish. And so this is what we started with at the beginning, was looking at Stellaris probes. And so these are singly labeled probes tiling across the mRNA. And this is well documented in the literature and was good to get working and worked great for microscopy, but it wasn't quite bright enough to do flow cytometry because we're talking about like 10, 15 molecules in these conditions. So we had to turn to a different probe technology from Affymetrix. When we bought it, they called it Quantifying. I'm not sure what they're calling it now. But essentially, it's this day-long hybridization where you build up this tree of oligos and then decorate that with fluorophores. And it took some doing to get it working in yeast. It's designed for mammalian systems, but we got it working. And so at the end of the day, you get these obscenely bright foci. Presumably, those are single mRNA, although we haven't checked that because we don't care about the microscopy. What we care about is the flow cytometry. And so here's just a distribution of GAP1 mRNA fish for wild-type cells starved from nitrogen. And this is with the probe, and in green, that's our on population. And if we omit the target probe that confers specificity, this is our negative, this is our background. And so if we add glutamine to these cells, we get a quick repression at 3 minutes and then a clearance at 13 minutes of of the signal. So this is pretty exciting, I think, that we can do mRNA fish with flow cytometry on like 10, 15 molecules, which is pretty cool. It's rather borderline, but it works. And so now we can go back to our system, right? We have our pooled library, and we've done a glutamine upshift, and we haven't, and we can now make them more red if they have more GAP1 mRNA. And now we can run them through a fax machine, and we can sort out. And what we're looking for is we're looking for mutants that are on, at the beginning of the experiment, and are still on, are defective in the ability to repress GAP1 mRNA after 10, 15 minutes of glutamine. And the way we're quantifying that is by doing bar-seq of the unique barcodes that are up and down of the tags in the deletion collection. So to quantify the mutants. And so this is what we did. Here's the entire deletion collection just by flow cytometry. This is our on population. And then you can see in the kind of the light blue there, or light green, is 14 minutes later. So most of them are off. And so what we wanted to do is find the stragglers that still have GAP1 mRNA on, are defective in the ability to repress it. And so here's just a ranked list of mutants that showed up in the top. And some of them make a lot of sense, but are perhaps a little bit more indirect. I'm just going to show you ASC1 here. Here's an ASC1 knockout compared with wild type. And this is just qPCR of GAP1 mRNA after the upshift. And so we can see it's defective in the ability to rapidly clear GAP1 mRNA subtly, but it is defective. And this is really curious because ASC1 is this it's really, really cool protein. And it's known to be required for translational arrest and cleavage of stalling or truncated mRNA constructs that are used to study things like nonstop mRNA degradation, no-go degradation, which is really curious. It's also been shown more recently to play a role in modulating ribosomal density of certain classes of mRNA, by recent work from the Gilbert lab. So it's rather interesting. And so what we think may be happening in the system is that we have GAP1 mRNA, highly expressed, ribosomes are tugging along, and what could be happening is, as was proposed, is that there could be translational changes. And so we're not sure how that works. We're going to walk through our list and try to find out what might be the mechanism for this. But what we think could be happening is that there could be a translation stall through specialized ribosomes or perhaps a aptamer or RNA binding protein or something like that that could be stalling translation, and then that could be activating these mRNA surveillance pathways that are co-translationally degrading aberrant, damaged mRNA. And so the thought is that we know that translational efficiency feeds back on mRNA stability during steady state. And perhaps it also is doing so in this transition. And what we see is accelerated mRNA degradation might not be for a particular purpose, but could be as a result of translational changes. So that's what we're looking to investigate and see if that holds water. So I told you about the destabilization of these NCR transporters during the upshift. I told you about the screen, the bar-seq after fax after fish. We've been calling it BFF in our notebooks. As a way to screen for wild type, for factors of mRNA dynamics. And I told you that at least one mRNA surveillance factor contributes to the accelerated degradation. And so with that, I just want to thank the folks in my lab who have made this possible, and especially past members Nikki and Benji. Also the great folks at NYU, a very tight-knit group, our collaborators, and also the great folks at the Cold Spring Harbor Yeast course, the instructors and assistants who make that possible. And with that, if there's any questions, I'd love to take them now.
If you delete components of the ribosome quality control complex, do you fail to get the glutamine effect? Because if you're causing a pause and it's an ASC1 mediated control, then it may be mediated by the ribosome quality control complex.
Yeah, so other things have come up. So what is interesting is, and kind of curious, is RKR1 or LTN1 comes up, which might suggest that this also could be indirect altogether. So we're curious and trying to figure out what's happening there.
Just a second, it doesn't have to be indirect at all. What you can have is some kind of pause that actually causes the stall, and then that's actually what's decreasing translation and transcription.
Yeah, definitely.
So it's kind of surprised that in your top hits you didn't find the TOR messenger RNA changes. So do you have a good explanation for that?
I'm sorry?
Because so in your top hits, that for those mRNAs, you found that the GAP1 and also the other transporters, but you didn't find the TOR target messenger RNA changes, because TOR is required for the amino acid signaling response, especially for the glutamine and the proline. So do you have a good explanation for that?
We haven't tested any mutants in the TOR complex 1 pathway as of yet, and we definitely didn't see any destabilization of TOR1 mRNA at all, or anything like that. Although we are also looking at, well, anyways.
Okay.
Yeah.
Okay.
But we haven't tested those mutants yet.
Have you looked at whether any positions in the GAP1 transcript are required for the fast degradation?
Not quite yet. So we're trying to think about how, like, where in the transcript, if it's maybe a cleavage at a particular position, maybe. We're trying to figure out what's going on with this. What we do know is that if we interrupt the 5' UTR, we don't get quite as quick of clearance, which is curious, and we're still trying to explain why that is. But we don't have a good answer for you yet.
One more quick question.
Thank you for the good talk. I have a technical question and a biology question. The technical part is I find the fish followed by flow cytometry and sequencing fascinating. And I assume you'll have to fix your samples first. And does that affect the sequencing afterwards?
I'm not sure. I don't know if it actually increases error rate. I haven't looked to see if it increases error rates in our sequencing, but I'd love to talk to you later about how we could possibly
That would
do
be great, thank you.
And about the actual methodology for getting these things to actually work with the fixation.
I'm actually a fly person, so I'm very interested in doing something similar, so that would be great to talk afterwards. And the biology question is, do you suspect a special RNA degradation pathway that's involved in this rapid degradation, for example, NMD or translational coupled degradation? Is there a specific one that you have in mind?
We checked a couple knockouts, and it looks like it's NMD.
All right, that was my guess. All right.
Yeah, okay, yeah.
Thank you. Let's chat afterwards.
Thanks.
Thank you.
Thank you, Derek. Thanks, Doc.